Welcome, everyone. My name is John Ludlow, and I am the Senior Scientific Advisor for Exosomes for ZenBio, a BioIVT company. And today, I'm going to present information and a talk on isolating, characterizing, and utilizing exosomes in research and in medicine, diagnostics, therapeutics. So to give just an outline of what I plan to cover today, first will be a brief background on exosome biosynthesis and their defining characteristics. Going to talk about the size, the protein composition, the microRNA, etc. And then I also want to cover the different isolation and characterization procedures and technologies that we use here at ZenBio and BioIVT company. And to compare that to perhaps what other companies or other technologies out there for isolation and characterization, but I'll focus on the ones that we use primarily, which are the result of our clients that we've had over the last 13 years of wanting certain analysis done a certain way, wanting certain isolations. Talk a little bit about the biological activity. And then again, as I mentioned earlier, we'll do some uses in some applications such as therapeutics, diagnostics, and one of our more recent adventures has been into the personal care products. I will then talk about scalable generation of adipose MSC exosomes, why it's important to scale these things up, particularly for the personal care products as well as other diagnostic uses. Quite a large number of exosomes needed to be generated for these purposes. And that's something you usually can't do with just typical tissue culture wear and tissue culture applications. We've also recently gotten interested in plant-derived exosomes, isolated from a variety of different plants. Most notably, these exosomes have been used in the development of personal care products. We also have some ready-to-ship custom isolations of exosomes, and we also focus a lot of our work on customized exosomes assays, again, depending on what the client needs. So a little bit about the biogenesis of exosomes. Typically, you have a cell and there's an endocytotic event. Something enters the cell where there's a pinching off of the cell membrane, the bilayer membrane, and then these membranes, these endosomes, will take up various proteins, nucleic acids, microRNAs, and then a lot of these will then become something called a late endosome or a multivesicular body that's shown down at the bottom of the slide there. These MVPs then have a choice, and there's a variety of mechanisms that determine which choice these MVBs make. I'm not going to go into those, but they can be destined for degradation in a lysosome, or they'll fuse with the cell membrane and dump the contents into the either cell culture media or the bloodstream, and these are the exosomes. So what happens after the exosomes are set free? Well, there's a couple of ways that they enter the cell. They can do it via endocytosis. We just talked about that a little bit. They will be endocytosed, which is an energy requiring event. And once they're inside the cell, then they again have two possibilities. They can then go to lysosomal degradation, or after the exosomes, after their contents are released, the RNA and the proteins, then they will be released in the cytosol, and then they will carry out their functions, whatever they may be. But we'll talk about those a little bit. There's also a possibility that the exosomes will go into the cell and then just come right back out again. I'm not going to spend any time on that. Fusion is another way where the exosomes essentially meld into the cell membrane. Again, the exosomal contents are then dumped into the cell. We call it cargo delivery. And then once into the cytosol, the proteins and nucleic acids will carry out their functions. There's also a receptor ligand interaction where there are specific receptors on the cell and the exosome, which the exosome will target the cell, fuse, and again, dump the contents and then carry out their functions. Some defining characteristics. Exosomes are a member of what they call extracellular vesicles. And really the big thing that separates extracellular vesicles, or EVs, from exosomes is the size. And there's certain specific proteins that are found on the membrane of the exosome. The three most prominent ones are CD63, CD81, and CD9. You also have ELIX, TS101, and HSP70. There are a couple others, but suffice to say, 63, 81, and 9 are the primary. They have been designated by the International Society of Extracellular Vesicles as defining characteristics. On the right-hand side of the slide, size is another thing. Exosomes are small, 30 to 150 nanometers in diameter. Next comes the microvesicles, which you'll see there is some overlap. Microvesicles can be 100 to 1,000 nanometers in size. And then you have the apoptotic bodies, which are very, very large, 1,000 to 500 nanometers. One of the things that we do to make sure that our population is biased toward exosomes is that all of our preparations are filter sterilized through a 0.22 micron filter, which means everything smaller than 220 nanometers will go through. So that really skews the isolation procedure for us to be primarily for exosomes. Some isolation methods. Again, these came about primarily from the requests by our customers. Ultracentrifugation has been the gold standard. That is how exosomes were first isolated. Virtually all of our clients, except for one, would like us to isolate by ultracentrifugation. The second most popular is size exclusion chromatography. In this table here, there's a list of pros and cons, advantages in time, etc. But suffice to say, other than ultracentrifugation and size exclusion, the ultrafiltration, immunoaffinity, and precipitation, these are all fine if you're looking for a small number of exosomes, say 10 to the 6th to 10 to the 8th. But when you want to isolate and you require more like 10 to the 12th, 10 to the 14th, these methods aren't going to suffice. The other thing that we've learned from our customers is anytime you have some sort of ligand on the exosomes, as you would get for precipitation, because it's usually some sort of bead. I'm sorry, the immunogenic separation uses a bead, but the precipitation, if it uses some other chemical to cause the material to come out, it usually attaches to the exosome, which can then influence downstream functions. For a lot of times, I don't know specifically what the ultimate function is for the customer, but they would rather that it not be tagged with anything. So they prefer ultracentrifugation or size exclusion chromatography. So our capabilities here at BioIVT. We do size determination by nanoparticle tracking analysis. That is also the gold standard, if you will, for characterizing these. NTA uses Brownian motion, and it essentially looks at the particles that go through a laser, and then based on the diffraction properties, which again is a component of Brownian motion, a size and quantity can be determined. We also do surface protein expression. We use the Miltenyi MaxPlex kit. When Miltenyi was developing this kit, which does 37 different surface proteins, they actually approached us to provide the exosomes to develop this technology and for comparative and standardization. RNA-seq is another characterization we do. Here's where you find out what the contents of the exosome are. And from there, you can glean putative properties or putative functions of the exosomes, based on their content and what they dump into the cell. Proteomics and lipidomics, again, surface protein analysis as well as lipid analysis. More and more now, the field is changing toward looking at lipids as a functional component rather than just the proteins. Historically, it was proteins, but lipids are starting to gain increased interest. Electron microscopy, that's another gold standard, if you will, to show that you have exosomes because you can see the size, you can see the shape. We also have the capabilities to Western blot for specific proteins of interest. Say you have exosomes isolated from a cancer patient and you're looking for a specific marker on the exosome, we can do that. Functional assays are another large component of the capabilities that we have and the services we provide. It's all well and good to have exosomes, but ideally, you want to demonstrate some sort of function. And I've listed several of them there. The most prominent ones that most of our clients ask for are cytokine production and collagen production, followed by cell migration. The others that are listed on there, we have assays developed, but the three I mentioned are the ones that are most prominently requested. Again, more of the same, but just here, I just wanted to point out the different cell types or materials that we've isolated exosomes from, the segments within the industry that these exosomes are used for, and then the applications, disease diagnosis, disease modeling, tissue regeneration, and again, more and more ingredients in hair and skincare products. On the far right there shows highlighted, but after you ultracentrifuge a biological fluid, you are able to visibly see the exosome pellet following ultracentrifugation. So when we take the supernate off, wash the pellet, and then we resuspend the pellet and do the quantitation and analysis. We have been in this space for about the last 13 years. Just wanted to show a handful of the publications that we've either written ourselves or have been invited to be co-authors on over the last 10 years. I think the first one was, or 9 years. I think the first one came out in 2016. Again, the key findings there, because each of the publications has a different goal or a different message that we want to convey. And so we have an extensive publication history in that area also. So getting on to characterization. As I mentioned earlier, expression of CD81, 63, and 9 are critical. Particle size, again, needs to be 80 or 30 to 150 nanometers. Most of ours run somewhere about 80 to 110, 120. Particle size and shape, bottom of the right hand, that's an electron microscope, photomicrograph. And you can see two exosomes there, one at about 9 o'clock, one at about 3 o'clock. The size bar at the bottom shows it's about 100 nanometers in diameter. And this was uranyl acetate staining, so it's going to be dark. But those are definitely exosomes that you see in the picture there. And as I mentioned earlier, we do surface protein expression analysis with the MaxPlex. On the bottom of the figure shows all the different proteins that we've identified. Pay particular attention to the three on the left, CD81, 63, and 9. These were the capture antibodies. So all the analysis was performed on particles that expressed at least one of those three proteins. And once we had those particles captured, then the analysis for the other proteins took place. And you can see there are a variety on there. A lot of them are cell cycle regulators. Some of them are adhesion proteins. It really just depends. Functional analysis. Spent a fair amount of time developing these assays. First and foremost was exosome-induced proliferation. When we started doing this, we were interested in wound healing. And one of the components of wound healing is to make sure that cells proliferate and they will divide and make more of themselves. So we took two different cell types, human keratinocytes and fibroblasts. Keratinocytes data is shown on the top, the fibroblasts shown on the bottom, and within each figure there, we show base media where there's no components added, or complete media, which in our case means there's serum. The thing about when you do these types of assays is that when you isolate exosomes, you have to make sure that there's no serum in the media, because bovine serum or human serum has a lot of exosomes in it. You can't accurately describe or test what the exosomes from your cells of interest are unless you're sure those other exosomes are not there. But then again, the exosomes themselves, to ask them to proliferate cells on their own is a tall order. So in order to show a differential here, what needed to be done was to basically back titrate the FBS and then add the exosomes and see if you see an additive effect. Um, and that is particularly shown in the right-hand panel there, which is a migration assay, not a proliferation, but migration. And focusing on panel B, that is our control medium. That is the one, or that is our vehicle, if you will. So everything else, or the millions of exosomes we added for D, E, and F, the 25, the 100, and 250 million, the 1% FBS medium was supplemented with those. And I think, as you can clearly see, comparing panel B to D, E, and F, these cells proliferate fine with the presence of exosomes. I'll stop short of saying that there is a pharmaceutical effect, or because it's not a stepwise growth, but you can still see that 25 million will do the job, as will 250 million. Additional functional analysis. This is a big one that a lot of customers ask for. Do these particles have the ability to reduce inflammation? And the short answer to that is yes. Left-hand side of the panel, the red bars are human-killed P. gingivitis bacteria. This bacteria is able to stimulate production of IL-6, IL-8, and MCP-1, they're inflammatory cytokines. You can see when we add exosomes from adipose-derived stem cells, the green bars, that these particles have the ability to reduce inflammation within these experiments, and even, if you will, outcompete or dampen the effect that the P. gingivitis has in stimulating inflammatory response. Collagen production is another one. Right-hand panel, we use TGF-beta as the positive control to show that, yes, you can increase collagen production. And here we actually seem to have a nice progression, whereas we increase the amount of exosomes, we see an increase in the collagen production. The last bar on that panel shows heat shock exosomes. That's another area that we've investigated of how can we tune the exosomes without necessarily doing any genetic manipulation. And what we found is if we heat shock cells and isolate exosomes from heat shock cells, that these cells contain a higher amount of microRNA and also protein, particularly HSP70 and HSP90. And again, these are protective proteins. And we seem to get an increased response in collagen production using heat shock proteins, actually 50% less compared to the standard exosomes, which weren't heat treated. So these two slides, exosomes have an anti-inflammatory activity and stimulate collagen production. Scalable generation. As I mentioned in the beginning, you can isolate exosomes from any biological fluid, or you can also isolate them from cultured cells. you're limited by the number of exosomes within you can isolate from cell culture, particularly if you have to grow them in tissue culture plastic, whether it be petri dishes, T75s, or Tendex. We've developed a method using commercially available bioreactors, which we purchased from FiberCell, where we can culture 1 billion cells on the cartridge. We are able to keep the cells alive for up to 3 months. Typically, we just go 2 months. And then we harvest the medium inside the cartridge, which contains serum-free media. Again, as I mentioned before, you don't want to have serum in the media if you want to test the function of your exosomes from your cells of interest. We can then pull off the media every day or every 3 to 5 days, keep it up for 2 months. And just to give you an idea of the scale, um, we will isolate over a 2 month period approximately 400 mils of conditioned media. And from that, we can isolate 10 to the 13th to 10 to the 14th exosomes. To put it into scale, if we wanted that many exosomes from Tendex, we would need approximately 20 Tendex, which would come out to somewhere about 20 to 30 liters of material. That's a lot if you're going to ultracentrifuge. It takes a lot of time. It's also not practical to have that kind of volume if you want to use column chromatography or precipitation. So again, this was a big thing for us and for our clients, particularly the ones that we supply exosomes for the cosmetic world, because 10 to the 13th, 10 to the 14th exosomes are needed for that type of production. So a couple of the key segment exosome markets. I've divided them into four here, just general purposes. First is EV engineering. There is a lot of work going on out there to modify the exosomes to enhance their therapeutic potential and their applications. As I mentioned, we decided not to pursue increasing function or changing the activities by doing genetic manipulation. We chose to do heat shock. But other ways to engineer these exosomes are to stably transfect the cells, or you can electroporate exosomes and put your nucleic acid or protein of interest in the exosomes. Therapeutic applications are focus on developing therapies that for targeting drug delivery, regenerative medicine, or treatment of various diseases. Many companies out there are particularly looking for ways to treat cancer. Along with that is the diagnosis. Companies are developing various technologies to identify specific disease-related markers on exosomes. And then the personal care. The exosomes is, again, I mentioned, based on the in vitro data, that yes, you can reduce inflammatory cytokines, you can increase collagen production, and these are things that are needed or accentuate skincare products to help reduce the appearance of wrinkles, increase luminescence, etc. Just want to go over a couple of limitations or things to consider when developing these products or for these specific areas. Exome engineering is at this point really limited. There's really not good targeting mechanisms. I mentioned specific receptors, but that work is lagging behind the ability to put drugs into the exosomes, and then the rapid systemic clearance too. You have to, and there are studies out there that indicate that this works, but you generally have to infuse exosomes into the circulatory system, and then they need to find their way to the diseased organ or the area you want to treat. The most success in this area has been around heart attacks, infarcts, since all the circulation in your body goes through your heart, also goes through your lungs and your liver. These are areas where a less targeted approach, i.e., a systemic approach, seems to have the best ability to treat specific illnesses. Vaccines and precision medicine, again, looking at tumor-derived exosomes, dendritic cell, and acetic cell exosomes. This area is also starting to take off with respect to, again, cancer and as well as some nerve issues. Lyophilization of exosomes is another area that we've actively engaged in because you want to do everything you can to keep the shelf life of these products as long as you can. Freezing, room temperature, and cold, yes, they will keep those, but sometimes you're not able to ship that way, particularly in developing countries where these items might be useful, or in the field. If there's a way to lyophilize them and still keep them active, that would extend the shelf life and actually make exosomes more versatile as a subunit of or a component of different treatments. Fluorescent exosomes, again, a great way to track where these things go, either in tissue culture or in animals. A couple of ways you can label them. There are lipophilic dyes out there that will enable fluorescent labeling of the outer coat proteins of the exosomes. And there's also dyes out there where you can label the microRNAs. This is particularly useful if you want to find out if the contents of the exosomes were actually dumped into the cell, because you can do time lapse and see that there'll be a fusion event or an attachment of the exosomes to the cells. And then over time, you'll see the fluorescent nucleic acid deposit and then disperse through the cytosol. So it's a good way to track. And then again, cell lines derived exosomes. There are a couple of companies out there that we have worked with that are developing cell lines that produce, they're hyperproduce exosomes. And a lot of times to get them to hyperproduce, it's a specific cell line and then a specific media with certain growth factors in it. And then having a cell line, again, then theoretically will give you an inexhaustible supply of this instead of having to go back to MSCs or fresh cells. Therapeutic applications, big challenges here. Even though there are over 200 clinical trials that have been registered with the NIH, it's still a challenge out there to meet the regulatory guidelines because these are so new. Is it cell or is it gene therapy? What's the cargo? Right now, it's a case-by-case approval, and any company that has a product they want to get through the FDA needs to go talk to them and needs to basically plead their case. And the FDA will listen. They want products to work, but they don't know everything. They will admit that. So if you're one of the first companies to get there and you can set the standard, that'll be great for the industry. Standardization. As I mentioned, there are multiple ways to isolate the exosomes. And you can get different subfractions, you can get in different amounts. So there needs to be, particularly for a therapeutic, some way of standardizing the manufacturing. A lot of times when you go to the FDA and you with a product, the method usually ends up being the product. So if you do all your pre-studies with exosomes that were isolated by ultracentrifugation, now you're gonna get a different composition of particles than you would by size exclusion chromatography. But if you stick to a method of isolation, show that it works in your particular degree of interest, then that will become the standard. Quality control. Batch consistency is a significant hurdle. Despite a lot of the best efforts that we've tried, you can standardize the number of cells, you can standardize the growth time. You will still get a different number of exosomes with a different size profile within a range. for the isolated products. The assessing the therapeutic efficacy is also a challenge. You really have to know what your endpoint assay is and is it relevant to your specific disease. And that's not an easy task, because along with that, you've got to understand what the molecular composition is and the structure. Where'd you get the exosomes from? Animal models for preclinical testing. There are a variety of animal models out there, but which ones are the best? Which ones are most representative for the therapy that you are interested in? It's also going to dictate your cell source for the exosomes, and the cargo that they have. So again, there are multiple challenges with therapeutic applications, but still, there are over 200 INDs out there. So it's going to happen. Diagnostic. I think many people have heard about liquid biopsy. That is a great way to be the least invasive method to get a sample for analysis for a disease state. The nice thing about exosomes is they are essentially a microcosm of all of the genes within cells in the body. So they're particularly useful for developing diagnostics. Now, here again, here's the difficulty in this. You have to know what proteins you're looking for. You have to know what RNAs, et cetera. Particularly in 2016 to 2020, there was a lot of diagnostic companies showing up and a lot of diagnostic applications being developed. But lately, for about the past 3 years, that seems to have shifted more toward a therapeutic approach than a diagnostic. The evidence for that is decreased publications in 2022 and a decrease in patents filed and issued for diagnostic versus therapeutic. It's not to say that diagnostic is a field that's going to go away, but seems to be more of a shift toward therapeutics than diagnostics. Personal care. As I mentioned, this is something that exosomes, EVs are gaining particular interest in. The MSCs are the ones that are usually skincare focused, anti-aging. Same with the adipose-derived stem cells. You can also use something called human platelet lysate or platelet-rich plasma. These biofluids are rich in exosomes, and there's been studies out there and products out there that tout that these exosomes from these sources can facilitate hair growth. Plant-derived exosomes, also an area where many companies are applying these exosomes to hair growth. In 2016, ZenBio had a licensing agreement with a company called Exosomes, where we provided isolated exosomes for inclusion into their skin moisturizer formulations. We then divested ourselves from that company and have sold the rights to another company, which are continuing to purchase isolated exosomes from BioIVT and continue to use them in their products. Plant exosomes. As again, I mentioned, they're starting to get more and more interest in the cosmetic world. And one of the reasons for that is there are some customers that are a little hesitant to want to have a human isolated product in products that they want to put onto their skin. And that's perfectly understandable. So we've chosen to look at the exosome activities that you can isolate from something like aloe vera. Aloe vera has been around forever. It's put on the skin to treat sunburn, a variety of things. So it's, if you will, it might be a safer alternative. And we've shown properties of, we've shown that they have properties to reduce inflammation and protect against environmental stressors. And I think that data is shown on the next slide. Yes. So we've characterized the plant-derived exosomes as we have the human and mammalian exosomes. We used nanodrop spec to look at protein content and look at protein quantitation and also RNA concentration, and the NTA to look at size. And as I've summarized on the bottom table there, these are exosomes that we isolated from a radish, granadilia, and a root. Um, the weights in there, how much we use for the isolation, the proteins, the RNA. And if you look at the end, you'll see that in the mode area, particularly, you were finding particles the same size as we'd seen in the for the mammalian cell exosomes, somewhere in the 80 to 120 area. But if you look at the different amounts there, some of these of the sample material, seem to have many more particles per unit weight than others. I mean, particularly with the granadilia there, I mean, there was barely enough there for us to measure, yet you got 10 to the 10th particles. Um, and then you look at the radish seed where we essentially got, for half the weight, we got 10 times as many particles compared to the root. So it really just depends on the source. We've also looked at these particles under EM, and yes, indeed, you can find exosome-like particles in the correct size range. Biological activity, we've shown a decrease in IL-6 and TNF alpha when cells incubated with aloe vera. Significant decrease in IL-8 and TNF alpha, and an increase in collagen production, which I've shown that data on the far right. And I think this is particularly telling if you look at the control there, which is TGF-beta, produces a large amount of collagen in this particular cell type. But both aloe and ASC at 6 times 10 to the 7th particles also stimulates the production of exosomes. I'm sorry, stimulates production of collagen. And in this case, the aloe seems to be greater than the human. And again, it just depends on the cells you take to do the analysis. But it's clear that even these plants' exosomes have a biological activity on human cells. In closing, then, I just want to say that at BioIVT, we provide high purity exosomes for research and development. We have ready to ship custom isolations. And the list there is from human cord blood, placental derived, and pre-adipocyte derived exosomes. Next slide. Custom assays. We collaborate with our sponsors to characterize, design, and basically do whatever type of analysis is needed. We have cell-based assays where, and we customize the cell-based assays to fit the requirements, whether it be a cancer cell type, whether it be a proliferation, or whether it be an angiogenesis type assay. We have all of those developed and currently working in-house. Characterization studies, this tends to focus more on the size, the molecular content, the microRNA, and also other specific proteins that they carry on their surface. Again, this is all client driven. So in closing, I just want to reiterate that BioIVT is a premier supplier of exosomes and services to investigate the exosomes. We have a 13-year experience characterizing and providing exosomes to scientists and commercial product developers. We've developed scale-up procedures, most notably for the adipose-derived stem cells, but the bioreactor is amenable to any type of cell that grows attached. We can put any types of cell in there from any source to isolate exosomes from. And finally, we have over 10 years experience in the personal care product development, and we are currently actively engaged with two other companies where we supply both plant and animal exosomes for use in cosmetic products, skincare, and hair growth. And with that, I thank you for your attention.